So today's video is eukaryotic gene regulation. The last one with the operons was prokaryotic. So let's focus on eukaryotic today. And we're going to look mostly at multicellular organisms. So organisms where cells need to differentiate. We've got, for example, think of us. You've got muscle cells, nervous cells, immune cells, blood cells, liver cells. You've got a wide variety of cells that do a wide variety of function. But these cells all have the same DNA. These cells are what are called totipotent, and that means that they are genetically identical. And so by genetically identical, we mean that they've got the exact same DNA in them, and the difference then is going to be which genes are actually being transcribed and what's being used. All of these cells, when they're totipotent, they have a full set of genes, and so they have the potential, what's called the total potential, to turn into any kind of cell. And again, that's going to depend upon the gene regulation, which genes are expressed. And of course, we always have an exception, and our exception here would be our white blood cells. White blood cells are not totipotent. They actually have they're different from one another in the antibody producing genes, so they can produce the unique antibodies to fight the different diseases. But the majority of most of multicellular organisms, the cells that are in there are going to be, like I said before, totipotent cells. Again, the differences will be due to their different gene regulation, which genes are being expressed. Errors in that can lead to cancer. Errors in the gene expression, the regulation of what's being transcribed. And so the regulation of that is controlled at a variety of stages. It's controlled in the replication stage, transcription, translation, and we're going to look at a variety of ways that these genes can be regulated as to what's being expressed and what's not. First of all, let's look a little closer at the DNA in a eukaryotic organism. Remember, the DNA in a eukaryotic organism is different than a prokaryotic organism in the sense that there's lots and lots of it. There's way more of the eukaryotic DNA than there is prokaryotic DNA. Remember, your prokaryotic DNA is just wound into that circle, or your eukaryotic DNA is wound into the chromosomes. And that is so 6 feet of DNA can fit into each one of your cells. 6 feet of DNA is coiled up into each one of your cells. So first of all, we've got the double helix formation. So the DNA is twisted in the double helix, and then that double helix is going to wrap around what are called histones. These are proteins, and you can see how when the DNA wraps around it, it looks like a string of pearls. So these histones, when the double helix of the DNA wraps around the histone, that'll form what's called a nucleosome. So the nucleosome are these beads that form when the DNA is wrapping around the histone protein. From there, those nucleosomes will then fold onto one another. They'll coil into what we consider the chromatin. Remember, the chromatin is that loose DNA. So those nucleosomes will wind into that chromatin. The chromatin will then form what are called the looped domains. The looped domains are built on a protein scaffold, and it's just another folding step. So we've taken these chromatin fibers, and we're folding them even more onto these looped domains. And then those looped domains, you can see they will fold on themselves as well to actually form then the chromosome, that form that is visible to us, that condensed, tightly wound portion that is found right before we do mitosis. And so that's how the eukaryotic cell is able to fit all of that DNA into each one of the cells. We also need to remember that eukaryotic DNA has a lot of non-coding DNA. Remember that junk DNA? For example, remember we have introns. Prokaryotic cells do not have introns. Those are those intervening sequences. That's where the intron part comes from. The intervening sequences between the exons that are being expressed. And then there's also a couple of different kinds of what are called repetitive DNA. And repetitive DNA is found in between genes. And so we have an example of what's called tandemly repetitive or satellite DNA. So your tandemly repetitive or satellite DNA, these are certain sequences of base pairs that are repeated over and over and over and again. These would be things like telomeres. Do you remember those on the end of the chromosomes? And every time that they're the ones that when the DNA replicates, remember how the DNA gets a little bit shorter every time it replicates? So a telomere would be an example of a tandemly repetitive or satellite DNA, certain sequences of base pairs that are just over and over, repeated over and over and over and over again. Okay? Another example of repetitive DNA would be interspersed repetitive DNA. And so your interspersed, sorry, I can't spell today. Your interspersed repetitive DNA, these are large segments of DNA, so big long segments of DNA that repeat throughout the DNA genome. And these are not necessarily identical. They're usually very, very similar, but not. They don't have to be completely identical. But they are repetitive. They are so similar that they are considered repetitive. And like I said again, large pieces of DNA that are just repeated throughout the DNA genome. So all of these introns, tandemly repetitive or satellite DNA, interspersed repetitive DNA, all of these are examples of non-coding DNA that you're going to find in eukaryotic cells. Okay, so we have all this DNA that's in the eukaryotic cell, and we need to start looking at ways then that we can actually regulate the DNA, what is being expressed from this DNA. So one of the ways that we can do that is we can actually alter the genome. Remember, the genome is all of the DNA, all of the genes that exist in the cell. And so if we're going to alter the genome, that could have an impact on which genes are being expressed. And so some of the ways that we can alter the genome is through what's called gene amplification. Okay, gene amplification is you do exactly what its name says. You amplify the gene. So, for example, you have some amphibians where there's over 1 million copies of rRNA that's made from this little particular set of DNA. So it's just a very high volume area. Okay. Another way that you could alter the genome is through gene loss. Okay. You could actually lose some genes. An example here would be what are called gall midges. That's a kind of insect. It's a little bug. Okay. And so the gall midges. Okay, so the gall midges actually start off with 40 chromosomes. Okay, they start off with 40 chromosomes, and all but 2 of their cells lose 32 of those 40 chromosomes. And the 2 cells they have that retain them are the ones that become gametes. So there's significant gene loss happening there. Okay, and obviously that's going to alter their genome. Okay, you can also transpose the genes. You can have transposition occur. Okay, and so with transposition, you're basically moving the genes around along the genome. The example they'll show you here and that I've got here in this picture is hemoglobin. Okay, and so you can see that the hemoglobin gene over time, it has been mutated, it has been transposed to other chromosomes, so it's been moved around to different chromosomes. And so it's got multiple copies, we'll use the term copies loosely, of the same gene because there's been mutations in those genes. Okay? But basically the genes have been moved around, which is going to have an impact on the genome and which ones are being, and the regulation, which ones are being transposed. Gene rearrangement is another way that genes can be regulated. The example we have here is antibodies. Okay? Remember antibodies. The genes that produce the antibodies. The antibody itself has 2 regions, basically a constant region. Okay, so what's called the constant region when that stays the same from antibody to antibody, and then what's called the variable region, and that's the one that's going to change, that allows the antibody to be unique for the various antigens. So during differentiation, the parts of that variable region will rearrange themselves. And so that's how we get unique antibodies for the various different kinds of antigens. So far we've looked at things that can rearrange the DNA itself, rearrange and alter the DNA sequence, and how those can impact gene regulation. But we also have a process that's called epigenetics. Epigenetics is going to be things that influence gene expression. So they have an impact on which genes are being expressed, but they do it without changing the DNA sequence. I didn't transpose genes. I did not rearrange them. I just having an impact on who's being expressed, but I'm not altering the DNA itself, the sequence of the DNA itself. And so we've got two examples of this. The first one being what's called acetylation. And you can see this happening in this picture here, the acetylation. So with acetylation, you're adding what's called an acetyl group. So you add the acetyl group usually to the histone. The example that you see here is their acetylation of the protein the amino acid lysine. Okay, so we acetylate the lysine that's on the histone. So that's on that little beat. Remember the DNA winds around the histone. So each one of these gray circles would be a histone. So when we acetylate the lysine amino acid that's on that histone protein, we add the acetyl group to that, and what it does is it decreases the affinity of the histones for both DNA and other histones. You see when the acetylation has happened, now these histones have separated out. They're not grouped together in this big clump. They've separated out, and the DNA is not wound as tightly as around the histone. And what that does is it makes it easier for transcription to happen. So when a section of DNA has been acetylated, it makes transcription easier because now there's easier access to the DNA to make that process happen faster and easier. The other example would be methylation over here, adding a methyl groups. And methyl groups is your CH4 groups. So adding methyl groups to the nitrogen bases, it can actually repress gene expression. So you add them to the nitrogen bases of cytosine. And you can see your cytosine, and it's called a CpG island. So we've got an area where there's cytosine followed by lots and lots of guanine. And so we add methyl groups to those cytosines, and that's going to actually repress or stop gene expression once that methylation has happened. So acetylation actually makes it easier for genes to be expressed. Methylation makes it harder. Methylation is also permanent. So if you have a cell that has methylated an area that has done methylation, if that cell goes through division, then that cell remains methylated, and the daughter cells that gene will still remain methylated. An example of methylation you see in what's called bar bodies on females. Bar body is the second X chromosome in the female, and so it is highly methylated. That second X chromosome is not transcribing its genes really. And when that is passed on, like we mentioned, the methylation is permanent, and so when that is passed on to the daughter cells, that is called genomic imprinting. And that genomic imprinting is going to keep those genes inactive. Okay, so let's shift gears and talk about transcription factors and how those can impact gene expression. So transcription factors are things that are going to bind to the TATA box. Remember that sequence, that TATA sequence that's in the promoter. So they bind to the TATA box, which again, remember that's in the promoter. And when they bind to the TATA box in the promoter, that's what's going to allow RNA polymerase to attach. So in eukaryotic cells, if there is no transcription factor bound to the TATA box in the promoter, then RNA polymerase cannot attach. So this is required for RNA polymerase attachment. And so once the RNA polymerase attaches, then you can actually get transcription of the gene. Upstream from the promoter, you may see what are called the proximal control elements. And so the proximal control elements, these are an inducer region. So these are an inducer region. So they're going to encourage the RNA polymerase binding here. to the promoter. So these help transcription factors bind the proximal control elements. Like I said, it's an inducer region that helps the transcriptors bind, transcription factors bind to the promoter. Even further upstream, you may see what are called the distal control elements. Okay, and the distal control elements are called enhancers, and these are really far away. Like they could be thousands of nucleotides away. You see how there's a little gap here in the DNA to make it short enough so you could actually see where those distal control elements would be. Okay, and so like I said, these are called enhancers, these distal control elements. And so the enhancer will work by, you'll have what's called an activator protein. Okay, and so the activator protein is going to bind to the enhancer. And so the activator protein binds to the enhancer, and that will actually bend the DNA back on itself so that the enhancer region, that distal control element, can attach to the promoter region. Okay, let's take a look at the picture that'll show us that. So if we look at this picture here, this will show us exactly how that's going to work here. Okay, so here's our activators way upstream here. Like I said, they could be thousands of nucleotides away. These are distal control elements. And so when the protein binds to the enhancer, so here's the little proteins, these little like things that look like the top of cups. Okay, and then the enhancer is actually on the DNA strand. So when the protein, the activator protein binds to the enhancer, you can see the DNA has bent on itself here. Okay, and so that distal control element will end up binding with the transcription factors and allowing the RNA polymerase to bind and then transcribe the gene. These activators and transcription factors are gene-specific. Okay, they are gene-specific. So let's look at an example of those. So what I have here are examples of two different genes. And remember, most of the cells in the body have the same DNA. And again, it's just which genes are being transcribed at any given time. So I've got two cells here. I've got a liver nucleus and a lens nucleus. So I'm in a liver cell and a lens cell. And so both of these genes exist in these cells. Okay, the albumin gene exists as well as the crystalline gene exists in both of these cells. But because the activators are gene-specific, the liver cell only has activators that will activate the albumin cell and albumin gene and not the crystalline gene. Where the lens cell has activators that will not work on the albumin gene but will activate the crystalline gene. So they are gene-specific. Talking about an example that uses these regulatory activator proteins in humans. Okay, we're going to look at what's called lactose persistence. Okay, lactose persistence. So the LCT gene. Okay, so we have what's called the LCT gene. This gene is going to produce an enzyme called lactase, and lactase is able to digest the lactose sugar. This is the sugar that's in milk. The LCT gene requires a regulatory protein that is coded for by the MCM6 gene. Okay, so the regulatory protein, so the activator protein that turns this gene on comes from this gene, the MCM6 gene. Okay, and so it produces this this activator, this regulatory protein that will turn on the LCT gene. That's going to make the lactase. Most humans actually stop making the regulatory protein after weaning. So basically, after they stop drinking their mother's milk, they stop making this regulatory protein. This MCM6 gene shuts down. Okay, and so they shouldn't, in theory, they shouldn't be able to digest lactose sugar anymore. But there is a mutation that can occur. That's what you see happening right here. This is where the mutation occurs. There is a mutation that can occur that allows this MCM6 gene to keep producing the regulatory protein. And so it keeps producing the regulatory protein that can turn that LCT gene on, and so those humans can digest the lactose sugar because they keep making lactase. okay, which officially is technically very few of us that are still producing lots of this lactase enzyme. You have to have this mutation to be producing the lactase enzyme. Let's look at another way that we can regulate gene expression. Okay, we have to be able to coordinate the expression of certain genes. For instance, we've got many biochemical pathways that are going to require multiple genes, but these genes are on different chromosomes, but they need to be transcribed and translated at the same time. Okay? And so this can happen at the same time because a lot of them will use, these will use the same activator proteins or the same transcription factors. Since they all use the same activator protein or transcription factor, as soon as that activator protein or transcription factor are present, then it will turn all of these genes on. The example you see here in the picture are sex hormones. Okay, remember the example we can see here in particular is testosterone. Remember, these are steroid-based hormones, so they have the ability to cross the cell membrane. So they enter the cell and combine with a receptor protein. And when it combines with that receptor protein, it becomes a transcription factor. And so that transcription factor is common to multiple genes. And so it can coordinate the pathway for development. So far we've talked a lot about ways that genes can be regulated before they're ever transcribed. Okay? With the transcription factors, activator proteins, rearranging the genes. So we're going to look now at a variety of ways that once they've been transcribed, we can still control them. Okay, so after transcription has taken place, we can still regulate them in some way, shape, or form. So these are post-transcriptional controls. And so one of the ones that we have is alternative splicing. Okay, and we've kind of touched on alternative splicing. We didn't call it this per se, but this is what happens after the RNA has been processed. We go through that RNA processing track where we add the poly A tail and the 5' cap, and we cut out all those introns. And when we remove those introns, remember we talked about how the exons could be recombined into different forms. Okay? And that's what your alternative splicing is that recombining of your exons, those genes that are going to be expressed, and so those different combinations can lead to different proteins. For example, let's look at your picture down here. Okay, this is from fruit flies, okay, and they have what's called a double sex gene. Okay, and so this double sex gene is this is the gene that is going to code for male or female development, and it can be processed two different ways. One way for females, one way for males, and it's going to depend upon if there is, it's called the trogene product. Okay, and so if the gene itself has 6 exons, okay, so you can see that here in the picture. And depending upon the presence of that trogene product, that's going to determine how those exons are linked together during that processing phase, once those introns have been cut out. Okay, so in females, the trogene is present, the product there is present in females, okay, and it's going to end up linking exons 3 and 4 together, and all the rest of them are deleted. Okay? In males, there is no trogene product, so it doesn't produce anything, and so it actually links together. Exon 3 is going to be linked to 5 and 6, and exon 4 is left out. Okay? And so that determines male versus female, again, based upon how this gene has been spliced back together once the introns have been cut out. A human example of this here, I'm not saying, we're giving you a lot of examples during this video. I'm not saying you need to memorize every single example. You do need to, though, always have an example in your back pocket that you know well that you can write about. Always need to have an example you can write about. Okay, so let's look at this human example here. Okay, so the example we have in humans is a calcitonin gene-related peptide. So this calcitonin gene-related peptide, this is a hormone. So here's my product here. This is a hormone that's it's made actually in the hypothalamus, and it's going to interact with the nervous tissue. So this is a hormone that is a vasodilator, It helps with pain transmission. Okay, so like I said, this is interacting with nervous tissue, and it's made in the hypothalamus. We also have, humans also produce another hormone called calcitonin. Okay, and so we've got calcitonin here on this side, and calcitonin is made in the thyroid gland, and it is going to raise calcium levels in the blood. So it's going to raise calcium levels in the blood when they're too low. It's going to pull them out of the bones, pull calcium out of the bones, put it back into the bloodstream. Okay? And this is done by alternative splicing of that CGRP. You can see the various combinations, the two different combinations. Once those exons have been spliced out, those introns have been removed, those exons have been spliced back together, and depending upon the combination will depend upon what is produced. We also have what's called RNA interference. Okay, so we're still in our post-transcriptional controls. So the messenger RNA has been transcribed, okay, but with this RNA interference, which is also abbreviated RNI, okay, basically what you have is parts of the messenger RNA, so parts of the mRNA, are tagged for destruction. And so if that messenger RNA is destroyed, then that's going to stop the production of the protein. Okay, we've got two basic types. Okay, we have what's called silence RNA or small interfering RNA, abbreviated siRNA, silence RNA or small interfering RNA, as well as what's called microRNA or miRNA. Okay, these are both examples, like I said, of things that do RNA interference. You can see that happening in the picture here. Okay, so we've got the microRNA that is bound with the target messenger RNA that's going to end up breaking down that RNA, and so then translation is inhibited. So we're not going to actually form the protein. Okay, we have a few more post-transcriptional controls, so after transcription, that we need to mention. Okay, we have chemical signals. Okay, those chemical signals are going to control what goes in and out of the cell. So you may have some inhibitors that don't allow messenger RNA to even leave the cell, and so it keeps some messenger RNA in and some messenger RNA is allowed to leave. So even though the messenger RNA was made, it doesn't mean that the protein will actually be formed, because translation won't happen if the messenger RNA wasn't allowed to leave the cell. Okay, so that's one of the ways that chemical signals could regulate messenger RNA, controlling their exit from the nucleus. Okay, we also have messenger RNA degradation. Okay, remember, these are going to be associated with that poly A tail and the 5' cap. Remember, one of their jobs was to help keep the messenger RNA from degrading too fast. And so the shorter that poly A tail is, as that poly A tail starts to shorten, that then allows other enzymes to come remove that 5' cap. And so now with the removal of the 5' cap, the nuclease enzymes are then going to be able to break down the messenger RNA. So the shorter the poly A tail gets, like I said, the 5' cap is able to be removed, and those nuclease enzymes can then break down the messenger RNA. Messenger RNA has a lifetime that can range from minutes to weeks. Okay? Another one is called ubiquitin. Okay? This is like a marker. Okay? This will come in and it'll tag the protein for degradation. And so once the protein is tagged for degradation, so you can see the ubiquitin here, okay, and so it's going to tag the protein, and once it has bound with the target protein and tagged the protein, that protein now is going to be broken down. Okay, a proteasome complex, I don't know if you can see that. Okay, a proteasome complex will come in and start breaking the protein down. Okay, and as the protein gets broken down, the smaller polypeptide fragments, those will be broken down even further by other enzymes, and that ubiquitin will be recycled. But this would be another way to regulate that messenger RNA being translated. And we have two post-translational controls we want to talk about. So we've made the protein. Okay, it's gone through transcription, it's gone through translation. We've got a protein here. So the first one that can regulate the protein and what's coming out of the cell would be modification of the actual protein. Okay, so modification of the protein product. And this is done with virtually all of the proteins. Okay, virtually every single protein, an example of this would be our methionine removal. Okay, methionine is our start amino acid. Virtually every single protein is going to start with methionine. But methionine isn't needed for the protein. It actually needs to be removed for the protein to become functional. Okay? And so that would be a modification of the protein product. We remove that methionine from it so that the protein is now functional. Another one would be our digestive enzymes. Digestive enzymes are proteins. Remember, all of our enzymes are proteins. Okay, and so digestive enzymes are usually made in a precursor form called zymogen. So they don't eat themselves. We need to prevent the digestive enzyme from digesting itself so that it will actually work on the food that we've taken in. And so the digestive enzyme is made in the precursor form of zymogen, and then once it's excreted, now that becomes functional. But it stays in its non-functional form until it's been excreted so it doesn't destroy itself. Okay, another example of a post-translational control would be adding a prosthetic group to the protein that's been finished. You can see that here, okay, in this picture that we have over here on the left. Okay, example would be hemoglobin. Okay, so hemoglobin needs to add a heme group to each one of the polypeptide chains. And so if heme is not available, so if this prosthetic group, this heme, is not available to be added, then actually the regulatory protein, okay, stops translation. Okay, a regulatory protein will inactivate the translation initiation factor so that translation won't happen anymore. So we don't have these proteins that won't be able to have this modification, that won't be able to get the prosthetic group added onto them. Okay, so we'll work with some models for this stuff coming up in class, okay, and we will go from there.